Hello, everybody, and welcome back to our monthly knowledge webinars. Today's topic will focus on the update to Annex 1 with specific focus on environmental monitoring of viable and non-viable systems. Welcome back if you are a returning participant, and if you're new, welcome to our platform. You'll find a lot of interesting topics as we go over every month on GMP and things with environmental monitoring. And also you'll find a lot of backlog on our Knowledge Center on our website. So I'll be your presenter, and my name is Jason Kelly. I've been with Lighthouse for the last 8 years. I'm the VP of Services, and I've been in this space for about 27 years working on environmental monitoring systems and in supply of the products and services around that. That's enough about me. Let's get into today's topic. But before we do, I would like to say I did mention our Knowledge Center on our website. You can get onto our Knowledge Center here, and you can go and look at all of the products, etc. And not only that, you can get great details about the different applications of our products. We don't just want to supply products, we want to supply solutions, and that's what we're all about. So welcome on board to all our newbies, and for our returning participants, I thank you for jumping on. So finally, after 14 years in the making, GMP Annex 1 was released on August 25th. So implementation for most of the new release is set for August 25th on 2023, with the exception of RABS isolators, which has a deadline set for 2024. There are a few items that stand out in relation to the implementation of the standard. The contamination control strategy. There's also discussions on quality risk management and environmental monitoring program. This will be the focus for the environmental monitoring. In the drafts of this annex, as far back as 2017, contamination control strategy emerged and became the next big shift in pharma contamination control. The contamination control strategy, or the CCS, is outlined as a governance of monitoring and controls. The CCS is a multi-element principle, and the method starts with a risk assessment and instills personal awareness within a quality culture. Contamination controls such as training, hygiene, gowning, the gowning process, designs, material selection, etc., equipment design, equipment cleaning and sanitization, facility utility designs, and facility utility design cleaning and sanitization are all developed based on risk assessments. SOPs and training and implementation of these controls are all validated. Monitoring of these controls involves personal, in-process, material, environmental, utility, even pest control monitoring. It is a facility-wide program which is governed under the contamination control strategy. And we have seen more technologies go digital, and all of this digital data converging into LIMS-based systems. The CCS will, with the digital data, will become a powerful strategy in a swift key decision-making on a facility-wide level. Let's take a look at the new PICS GMP Annex 1 and how particle monitoring for air, microbes, and liquids are impacted, and how today's technologies can help meet the new guidelines and assist you in the transition to a CCS. We'll be focusing on one section of the CCS, and that is on environmental monitoring. So let's start off with a basic question. How big is a micron? ISO 14644 Part 1 refers to particles smaller than 0.1 micron as ultrafine particles and particles larger than 5 micron as macroparticles. In manufacturing processes, particles larger than 100 microns and smaller than 0.01 microns are of little interest, as particles larger than 100 microns are easily filtered, and particles smaller than 0.01 micron are so small to cause damage. I'm talking about like in the hundreds of nanometers. However, as technologies improve and semiconductor and biopharmas go smaller and smaller, then ultrafine particles become of interest. I was like, let's have a little fun and do a little experiment. And if you can pull out one of your hairs, I know this sounds funny, but if you can, pull it out and look at it. And look at the diameter of your hair. You know, we talk about environmental monitoring and particle counting, but let's just go right back to basics here and let's look at what we're trying to see. You know, a human hair under a microscope is about 80 to 100 microns in diameter. And here we have a nice little graphic here of human hair and how it looks against like a salt grain, which is about 60 microns, all the way down to viruses, which are 0.1 microns. And then we look at bacteria, which is the main concern in the pharma industry, around 5 microns. So you can see that we really have a challenge in what we do, and we really are blind without particle counters. And I just wanted to make that point. So let's just go into the environmental monitoring section of the new Annex 1, and I'm going to look at the general, and it's a separate chapter for environmental monitoring, and it's called Environmental Monitoring and Process Monitoring. In the new release, there is a clear differentiation between qualification, which includes classification, and monitoring. The site's environmental and process monitoring program will form part of the overall contamination control strategy and is used to monitor the controls designed to minimize the risk of microbial and particle contamination. And that's what they're saying. They really want to focus in on the environmental process and the monitoring and the programs involved. So it's very important to be understanding what you're trying to monitor. And it should be noted that the reliability of each of the elements of the monitoring system, I'm talking about the viable, the non-viable for the aseptic process, when taken into isolation, it's limited and should not be considered individually to be an indicator of failure of the process, the aseptic process. So there's multiple devices that are being used. The particle counters will detect particles and air samplers and settle plates, and then we've also got rapid micro coming into play here. They will detect if there's a biological presence. But definitely with the larger particles of around 5 microns, they can be a avenue for biological particles to fall onto or get into the product at the end of the day. So it's really interesting that it seems like a more harmonized approach will be used using multiple monitoring techniques and not relying on data from one technique, but to combine all data when making a decision. And this is a pretty wise move, as we sometimes see that high particle counter data does not necessarily mean that there is a high probability of viable contamination. There's no correlation. It would be safe to say that if you get particle counts exceeding threshold limits and 0 CFUs for the same location and time period, then the end result does not have a probable effect on the product's integrity. However, with an established environmental monitoring program and appropriate risk assessment, then the quality of data and the decision-making becomes more reliable. The new update states that the monitoring program should be considered in the overall context of the trend analysis and the CCS for the site. That's the contamination control strategy. Actually, I'm going to make it easier. I'm just going to call it a CCS from here on. So just to reiterate, the aseptic process simulation is a simulation of the entire aseptic manufacturing process in order to verify the capability of the process to assure product sterility. And I need to just talk about this in a little bit of detail, that the simulation, it's really wise that you do a simulation of your system and your actual facility running as it should, and then you can with that simulation and with the data that you're capturing, make sure you include all aseptic operations associated with routine manufacturing, equipment assembly, formulation, filling, even lithophilation if you're going to be doing that in the sealing process as necessary, and the validation that monitoring controls are in place and functioning as expected. That's important. We need to validate that. So we will not be covering that section, as it would take a whole webinar on the topic, but I do encourage you all to be familiar with this section here, as there is some useful information contained within, especially if you work in a full finished aseptic process environment. So the new Annex 1 says that an environmental monitoring program should be established and documented. The purpose of the environmental monitoring program is to provide assurance that clean air equipment continue to provide an environment of appropriate air cleanliness in accordance with design and regulatory requirements, and effectively detect excursions from environmental limits, triggering investigation and assessment of risk to product quality. Risk assessments should be performed in order to establish this comprehensive environmental monitoring program, i.e. sampling locations, frequency of monitoring, monitoring methods, and incubation conditions, such as time, temperatures, aerobic and anaerobic conditions as well should be all considered. It's a real harmonized approach. So these risk assessments should be conducted based on detailed knowledge of the process inputs and final product, the facility, equipment, the criticality of specific processes and steps, the operations involved, the routine monitoring data, the monitoring data obtained during qualification, and knowledge of typical microbial flora isolated from the environment. The risk assessment should include the determination of critical monitoring locations, those locations where the presence of microorganisms during processing may have an impact upon product quality, for example, in your grade A aseptic processing areas and the grade B areas that directly interface with the grade A areas, etc. Let's look at the rules specific to environmental monitoring. To make it easy for you all, each of the rules in the yellow boxes will be numbered as they are found in the guideline, and each slide heading in this specific section title will, you'll find this in the guideline. So as I'm saying, as we progress, I will take out the actual contents from the environmental monitoring guide, and then you'll be able to correlate them with what we're discussing here in each slide. So also, before I forget, you can download the guideline here for free. The download link is in yellow down below on this slide. And I recommend that you do download it and have a good look over the whole Annex 1 guideline. So let's start off and look at the environmental and process monitoring. And the routine monitoring of clean rooms, clean air equipment, and personnel should be performed in operation throughout all critical stages of processing, including equipment setup. I recommend, if you're not doing so already, to monitor the pre and post processes, not just your process. So monitor the pre-assembly setup should indicate that the environment meets grade A cleanliness. I would say at a minimum collect 1 cubic meter sample volume so you can show you meet grade A cleanliness levels. The same is true for post-monitoring. When the production ends, I would gather that a minimum another cubic meter sample. So for the process monitoring, I would use trend analysis based on 1 cubic foot 1 minute samples from your particle counters and using X out of Y events rather than following the certification classification particle concentrations based on a cubic meter. Some company using a continuous monitoring system will use 1 CFM particle sensors and run for 36 minutes so they capture a cubic meter sample. Again, I would just make sure that you are capturing these events and monitoring an adequate volume of air. And if you need further information on that, you can definitely get back to me, and I can go further and deeper into that topic with this 9.5. If you look at 9.6, other characteristics such as temperature and relative humidity should be controlled within ranges that align with product processing, personnel requirements, and support maintenance of defined cleanliness standards, e.g. grade A and B. I would also recommend and strongly recommend that your facility has a continuous environmental monitoring system with temperature, relative humidity, and also differential pressure sensors, which are independent of the building management system. And these sensors are for control in the BMS, and the EMS sensors are for the validated data. EMS systems are validated according to CGMP standards and GAMP. So I've seen cases where a facility is using 1 sensor for control with their BMS and monitoring and using that sensor data for batch release. And the FDA really comes in and says that's a nope. They do not want to see 1 sensor for control and also using that data for validation. You need to have separate sensors. And I've seen facilities where the customer has had to add in the extra sensors whenever the FDA or any other regulatory body, and actually over in Australia, we had the regulatory body over there and a couple of companies nearly shut down production because of that. And we went in and retrofitted a couple of environmental monitoring systems with the correct sensors and types independent of the BMS. So here's a fun thing to consider. What if you were using 1 sensor and that sensor drifted? How would you know, if you have one sensor. So BMS sensors are not routinely calibrated, so that drift could go on unnoticed. But if you had an independent sensor monitoring the same location, that drift would be caught pretty quickly. So overall, room temperature should remain at an ambient for personnel comfort. Anything above 25 degrees Celsius and with some moisture in the room could enable a suitable breeding ground for viable particulates. So again, that's just an example of the drift in electronic sensors, and not having an independent to watch. You may not actually see that drift happen. It could happen gradually over time. So definitely you want to monitor for TRH and DPs continuously, and I'm talking about 24/7 in your clean room with your environmental monitoring system, and add that into your environmental monitoring program. Moving on, still under environmental and process monitoring, the monitoring of grade A should demonstrate the maintenance of aseptic processing conditions during critical operations. Monitoring should be performed at locations posing the highest risk of contamination to the sterile equipment surfaces, containers, closures, and product. The selection of monitoring locations and the orientation and positioning of sampling devices should be justified and appropriate to obtain reliable data from the critical zones. Here we see an example of a grade A filling machine which has 4 locations to monitor in the grade A environment. We call these locations critical zones. We see location 1, the accumulator, has pre-washed open vials, so this would be deemed a critical location to monitor as the vials are open and at high risk. Location number 2 monitors the transport of the open vials to location number 3, the fill head, where the vials are filled with sterile product, before going to the stopper capping location at number 4. Sampling methods should not pose a risk of contamination to the manufacturing operations. This is section 9.8. So the material of construction, the ability to clean and sanitize the measuring equipment should be assessed. For example, the portable air sampler should have a HEPA filter exhaust. The base plate and sample head should be easily removable for the autoclave. Interface should be intuitive, and a touchscreen is more desirable rather than having buttons and more switches to press. The design of the unit should not allow the introduction of particle traps. Buttons, switches that stick should be avoided. Sample heads and bases for continuous samples should be stainless steel so they can be easily autoclaved. And here we have 2 examples, a portable air sampler and a sampling head which attaches onto a continuous viable system. So section 9.9, and this is a good one, and I want to spend a little bit of time on this. And we've actually, on our knowledge center, and I bring this up because you can go to our knowledge center and you can find that paper that I'm going to talk about under particle count strategies on alarm settings. So 9.9, appropriate alert levels and action limits should be set for the results of viable and total particle monitoring. The maximum total particle action limits are described in table 5, and the maximum viable particle action limits are described in table 6. We'll get to them a little bit further. However, more stringent action limits may be applied based on data trending, the nature of the process, or as determined within the contamination control strategy. Both viable and total particle alert levels should be established based on results of clean room qualification tests and periodically reviewed based on ongoing trend data. So this is a critical one to get right. Appropriate alert and action limits. That statement there is the key to success. I do have a whole paper, as I said, it's here in our knowledge center that you can download that discusses this topic in full detail, even with examples. So if you do go to our website and click on the knowledge center tab, sign up for the free access to this paper with some work examples on the trending of the data and setting the alarm limits. Overall, I would highly recommend you put some good energy into this and base your alerts and action limits on trending rather than following the table based on classification. As I said in my opening, the Annex 1 does clearly differentiate between monitoring and certification. So the issue with, and I want to talk, the issue with adding alarm limits based on the table for clean room classification is well documented and basically turns into a non-event, but costing in some cases several thousands of dollars or tens of thousands or hundreds of thousands of dollars to investigate the root cause. And what you're doing is when you do have the alarm exceeded based on the classification tables, you're looking at a one-off event. That's 1 minute, and you could have 4 hours of zero counting one event that will trigger an alarm, and then 4 hours of zero counting again. What's the probability of that being a biological event, an excursion that can have a detrimental impact on the product? What's the probability of that one event getting into those little vials and the diameter of the vial? So you got to think about the whole thing from a holistic approach. And I really do encourage setting your particle count strategies on alarm settings based out of trending and X out of Y events. And that paper that I've discussed here on our website will definitely give you some good insight. So 9.14 under environmental monitoring for total particle, and we're getting into the section on particles. So a total particle monitoring program should be established to obtain data for assessing potential contamination risks and to ensure the maintenance of the environment for sterile operations in a qualified state. So the monitoring program should consist of a continuous monitoring system. And here we see the grade A filling line with the 4 locations being monitored in the accumulator, the transport belt. And remember, the glass vials are open and empty at this stage. Then we have the fill head, where the sterile product is filled into the vial. And finally, we have a stopper and capper location. The total particle monitoring should monitor these critical locations along with background locations and any other locations where the product goes in and out of the clean room. So remote particle counters are used for this type of monitoring. So in this image, the samplers are taken via isokinetic sample, and the sample probes, which are made from stainless steel, are connected to the inlet of the remote particle counter. We got an example of a remote particle counter here. So look at section 9.15, the limits for environmental monitoring of airborne particle concentration for each graded area are given in this table. This is one of the tables I spoke about earlier, and you can find this under environmental monitoring total particle. And as we can see, the limits here for grade A, B, C, and D are outlined for at rest and in operation conditions for 0.5 microns and for 5 microns at a sample volume of a cubic meter. So we see that the 5 micron data in grade A, an environment is based per cubic meter sample, and the limit is 29 particles for both at rest and in operation environments. For 0.5 microns, the concentration limit per cubic meter is 3,520 for both the at rest and in operation stages. For grade D, in operation limits are not predetermined. The manufacturer should establish in operation limits based on a risk assessment and on routine data where applicable. So under the notes, which I don't have shown here, note 1, the particle limits given in the table for the at-rest state should be achieved after a short cleanup period defined during qualification, and guidance value of less than 20 minutes in an unmanned state after the completion of operations. And what they're saying here is to make sure that the aseptic area is ready to produce, to do the aseptic processing, and that you've met the criteria in the table here before you actually jump into the actual processing. Note 2, which isn't shown, but I'm going to talk about it, the occasional indication of a macro particle counts, especially greater than equal to 5 microns within grade A, may be considered to be false counts due to electronic noise, stray light, coincidence loss, etc. However, consecutive or regular counting of low levels may be indicative of a possible contamination event and should be investigated. Such events may indicate early failure of the room air supply filtration system, equipment failure, or may also be diagnostic of poor practices during machine setup and routine operation. And this is an interesting statement, which really talks and directs you towards trending, rather than just following, this table and having a one event where you have 1 minute sample, as I outlined earlier. You could have 4 hours of zero counts, one sample in the 1 minute, which would exceed the alarm limits here, and then you could have another 4 hours of zero count. That's not a trend. That's an event, a transient event. So what we're looking here is for trends and failures in the system. And again, I can't reiterate and highlight and really state this anymore. Your alarm limits are very important, and you setting them up is very important. And what I'm saying is not to follow the certification tables, but to use an X out of Y trending event scenario, which is described in that paper I've mentioned in the Knowledge Center. Section 9.16, for grade A, particle monitoring should be undertaken for the full duration of critical processing, including equipment assembly. As outlined in the previous slide, the equipment assembly stage should be monitored to ensure grade A conditions are maintained. Here we see a cleanroom technician inserting Petri dishes into a continuous air sampler, and the particle counter continuous system should be monitoring this assembly process. So I can't really see it on this diagram that there is a particle counter sampling head nearby, but definitely I would, in my risk assessment, I would put one in that location to validate that this process is not causing any issues with contamination. Again, because it's inside an isolator, then you're minimizing that effect because the main contributor of the particles, the human, is on the outside. But if you can imagine this was an open grade A environment, then you would have some concerns about the operator's interactions, especially with swapping out the media dishes into the air sampler. Section 9.17, under environmental monitoring under total particle, we're still under the total particle section. The grade A area should be monitored continuously for particles greater than or equal to 0.5 and greater than or equal to 5 microns, and with a suitable sample flow rate, at least 28 liters, 1 cubic foot per minute, so that all interventions, transient events, and any system deterioration is captured. The system should frequently correlate each individual sample result with alert levels and action limits at such a frequency that any potential excursion can be identified and responded to in a timely manner. Alarms should be triggered if alert levels are exceeded. Procedures should define the actions to be taken in response to the alarms, including the consideration of additional microbial monitoring. Again, all interventions, transient events, and any system deterioration is captured, and the system should frequently correlate each individual sample result with alert levels and action limits at such a frequency that any potential excursion can be identified and responded to in a timely manner. So again, it really is about employing a environmental monitoring system using your particle counters and also using microbial samplers as well and automated systems. Again, you won't know with the microbial at this stage because it will take up to 72 hours, but your particle counters are good indicators. And again, you can start implementing some of the rapid micro technology into this as well to try and speed up the process. Section 9.18, it is recommended that a similar system be used for the grade B area, although the sampling frequency may be decreased. The grade B area should be monitored at such a frequency and with suitable sample size that the program captures any increase in levels of contamination and system deterioration. If alert levels are exceeded, alarm should be triggered. Yes, I agree. On this, I would even go as far as to say that, if you're implementing, 4 particle counters in your filling machine and you got a couple of background areas, well, I would just let those, grade B sensors run at the same frequency as the grade A. And even though the guideline says the sample frequency may be decreased, I'd say further down that guideline, it calls for continuous monitoring of grade B, as we'll see, as you need to capture any increase in levels of contamination, and you can only do that by continuously monitoring and trending of the data. So this section, to me, is a little contradictory. However, in practice, the norm in the pharma industry especially is to continuously monitor grade B locations. And here we see locations 6 and 5 being monitored. And just for your information, when I use the term continuous monitoring, I'm referring to the monitoring continuously throughout the aseptic fill process, including the assembly stage. It's not 24/7 monitoring, only during aseptic production. But I would recommend, as I said earlier, 24/7 monitoring of temperature and relative humidity and differential pressure. Again, DP is critical. If differential pressure is lost within the rooms, then contamination risks from lower grade rooms is very probable. But getting to the section 9.18, I would actually recommend that you monitor your grade B background areas that are supporting grade A at the same frequency as the grade A. Section 9.19, the selection of the monitoring system should take into account any risk presented by the materials used in the manufacturing operations. e.g. those involving live organisms, parity products, or radiopharmaceuticals that may give rise to biological, chemical, or radiation hazards. So again, the selection of the monitoring system is critical. The sensor type and design can impact here. I recommend you look for a sensor that has a sealed enclosure and all vacuum and communication cables are covered, like in this example. And this example shows a nice sloped top cover. And then also you can see that, or you can't see, there is no vacuum tubing exposed or communication cables. And where hazards can present a danger, then a monitoring plan can mitigate those hazards. For example, the monitoring can be before or after the processing, so the equipment is not exposed to hazardous materials. And this is acceptable with an appropriate risk assessment and meets CGMP. And the next guideline agrees with that, with what I just mentioned here. Again, it says here, in the case where contaminants are present due to the process involved and would potentially damage the particle counter or air sampler or present a hazard, the frequency and strategy employed should be such as to assure the environment classification both prior to and post-exposure to the risk. And increased and viable particle monitoring should be considered to ensure comprehensive monitoring of the process. Additionally, monitoring should be performed during simulated operations. Such operations should be performed at appropriate intervals, and the approach should be defined in your contamination control strategy. And here's the acceptance is outlined for less frequent, non-viable monitoring, but you can mitigate with increased viable monitoring. A continuous air sample may be employed, but you must ensure the sample there is recirculated back into the scrubbing filtration of that biosafety cabinet or the radiopharmaceutical cyclotron. And the sample there should not make any contact with the cleanroom text in and around the processes environment. So safety first, and also you want to make sure that the products are safe from the exposure. For example, some particle counters may not work well in radiopharmaceutical cyclotrons because they may get false positives from the actual bombardment of those radio frequencies. Something just to be aware of. If you look at section 921, still under total particle in the environmental monitoring stage, the size of monitoring samples taken using automated systems will usually be a function of the sampling rate of the system used. It is not necessarily for the sample volume to be the same as that used for formal classification of clean rooms and clean air equipment. Monitoring samples volumes should be justified. I often see a lot of confusion regarding this one. It also adds confusion regarding setting alarms, as some people interpret the guideline incorrectly and apply sample volumes of a cubic meter into continuous monitoring systems. Here we see a volume of a cubic meter, and then we look at the volume of a cubic foot. There's a huge difference between the two. And the sample rate of a remote particle counter is typically 1 CFM. And we've seen in the guideline number 917, the exception is that the remote particle counter should sample 1 CFM. And the sample rate is based on 1 minute intervals. So as long as you use 1 CFM remotes, then you are within expected practices. And now I'm not saying that 0.1 CFM remotes may not be used. If you do use them, then you will need to justify them. You're taking less of a sample with 0.1 CFM. And you usually find 0.1 CFM in handheld units. There are some remotes that are available with 0.1 CFM. And just for your information, the ratio of 1 CFM to 1 cubic meter is about 35.4. So a cubic foot is 35.4 times smaller than a cubic meter. So with a cubic meter, you need to take a hell of a lot more of a volume. But as it says in this section 921, the size of monitoring samples taken using the automated system, the continuous environmental system, and it's usually a function of the sample rate. And down around here, the remote particle counters are sampling at 1 cubic foot per minute. Now we're going to move on to environmental and personnel monitoring, viable particle. And it says where aseptic operations are performed, microbial monitoring should be frequent using a combination of methods such as settle plates, volumetric air sampling, glove, gown, and surface sampling. Example, swabs and contact plates. The method of sampling used should be justified within the CCS and should be demonstrated not to have a detrimental impact on grade A and grade B airflow patterns. Clean room and equipment surfaces should be monitored at the end of an operation. As I said, now we move on to the viable particles section, focusing on personnel and the environment. And this guideline outlines the equipment used to capture viable contamination. And we see here there's several methods. We have the swabs, the contact plates, the settle plates, and the active air sampler there on the right as an example. So 923, viable particle monitoring should also be performed within the clean rooms when normal manufacturing operations are not occurring, e.g. post-disinfection, prior to start of manufacturing, on completion of the batch and after a shutdown period, and in associated rooms that have not been used in order to detect potential incidents of contamination which may affect the controls within the clean rooms. In case of an incident, additional sample locations may be used as a verification of the effectiveness of a corrective action, e.g. cleaning and disinfection. So, we talk about portable air samplers are very effective in this criteria, and they should be used for this type of monitoring. And I would recommend that the selection of this type of air sample should be considered in the context of ISO 14698, which has been shelved, and the more current standard is EN 17141. There is a new ISO standard under draft at the moment. It's not completed, and I believe that is ISO 14644 Part 20, which replaces the ISO 14698. I highly recommend that you check out these standards. Again, you can find these standards in our Knowledge Center, and there are technical papers written about these standards. But effectively, what this section is talking about is the use of additional monitoring to verify that rooms that are adjacent to your main core aseptic processing are frequently monitored so that you are not picking up any type of contamination and bringing it into the aseptic process core. So that's really important to understand that, and you can achieve that with these small, easy to use portable air samplers. Now we talk about continuous viable air monitoring. So 924, continuous viable air monitoring grade A, example air sampling or settle plates, should be undertaken for the full duration of critical processing, including equipment, aseptic setup, assembly, and critical processing. A similar approach should be considered for grade B clean rooms based on the risk of impact on the aseptic processing. The monitoring should be performed in such a way that all interventions, transient events, and any system deterioration will be captured, and any risk caused by interventions of the monitoring operations is avoided. Again, we're moving on to, and this is an important clause, we're now talking about continuous monitoring during the process. Before, in the old days, you could get away with sequential monitoring, you could get away with pre and post. Here they're really talking about continuous viable air systems. And these continuous viable air systems are available. And as I said, this is a major change, and I would recommend going for a lower flow rate sample. And so the frequency of Petri dish agar plate change rate does not impact on the process with too many operator interventions to change out the dish. This will be quite a challenge for industry to overcome, and as there may be many challenges to get the system running without it causing issues, where operator interventions to change out the air sampler media may be a major issue. Again, that comes down to training the operators, ensuring that they're meeting aseptic conditions, their wrists aren't exposed, the gloves are, put on correctly, the face masks are worn correctly, and they're not hovering over the process. So I would say watch this space, as the guideline does say use of active air samplers or settle plates. So maybe one solution could be a combination of the two technologies working side by side. It'll be interesting to see how this guideline will be interpreted, but it does say active air sampling. Here we see an example of a continuous active air sampling system where you can remotely start and stop the sampling, and all that data will go to the environmental monitoring system, where it will capture the location, the volumes that were sampled, who sampled, the time duration, etc. So again, this is coming hard and fast, folks, and I just want you to be prepared for this change. Section 9.25, a risk assessment should evaluate the locations, type, and frequency of personnel monitoring based on the activities performed and the proximity to critical zones. Monitoring should include sampling of personnel at periodic intervals during the process. Sampling of personnel should be performed in such a way that it will not compromise the process. So, particular consideration should be given to monitoring personnel following involvement in critical interventions. At a minimum, gloves, but may require monitoring of areas of gown as applicable to the process, and on each exit from the Grade B cleanroom, gloves and gown may be monitored. Where monitoring of gloves is performed after critical intervention, the outer glove should be replaced prior to continuation of the activity. And this is just something that you're all aware of during your process, or you should be. And now where the monitoring of gowns is required, after critical interventions, the gowns should be replaced before further activity in the clean room. So what we're talking about here is more awareness of these interventions and the impact of these interventions. And this goes back to operator training and SOPs being put into place for your contamination control strategy. So there's a lot to consider in this Annex 1, folks, and it's really coming hard and fast. Luckily, we have until 2023 to get it right and to get all this in place. So again, it's good that you're on board here to see the impact. And we're just looking at the environmental monitoring for viable and non-viable. The whole annex covers the whole facility. So it's definitely worth jumping on and making yourself aware of what's coming. And be prepared for the change, because the inspectors and auditors in 2023 in August are going to expect you to have these criteria in place. Section 9.26, microbial monitoring of personnel in the Grade A and Grade B areas should be performed where operations are manual in nature, e.g. aseptic compounding or filling. The increased risk should lead to enhanced emphasis placed on microbial monitoring of gowns and justified within the contamination control strategy. As we are all aware, folks, we are the main vessels of viable contamination in the cleanroom. I could see a good case for using rapid microdetection technology where operators are gowning up, or at least in a simulation exercise, to see how effective the gowns and the gown up procedures are in reduction of viables between being carried into the clean room. There are other technologies that we've worked with when it comes to clean room gowns being tested. So if you're needing any recommendations on that process, please let me know and I can go deeper into that topic. Just be aware that there will be more emphasis on personnel and clean room garment monitoring and testing validation, and SOPs should form part of your contamination control strategy and your overall facility plan. Section 927, where monitoring is routinely performed by manufacturing personnel, this should be subject to regular oversight by the quality unit. Aseptic operations, including, let's say, APS, for example, should be observed on a regular basis by personnel with specific expertise in aseptic processing to verify the correct performance of operations, including operator behaviors in the clean room, and address inappropriate practices if detected. I believe cleanroom operator behavior may be monitored via security cameras placed inside cleanrooms, as I'm seeing more new facilities come online with cameras everywhere in the cleanroom. As I am intrinsically opposed to Big Brother in general, I know we're feeling like it's 1984, folks. I do feel that the fact that you are aware that you're on camera, I think cleanroom operators will think more about their movements in and around the cleanroom. And if you ever sit back and observe people in the cleanroom, it's really important to observe their movement and understand the airflows. And even just raising your hands above, you are changing the overall airflow of that room by your movements. So you don't want to move too fast. You don't want to wave your hands around. You don't want to do any jumping jacks or dancing. I've seen some videos of people dancing in the clean rooms. They're quite funny, but in reality, you are causing a lot of issues. You're causing more harm than good, folks. And at the end of the day, we want to, make sure the patient is protected from contamination, and to consider your actions in and around the clean room. Section 928, the adoption of suitable alternative monitoring systems such as rapid methods should be considered by manufacturers in order to expedite the detection of microbial contamination issues and to reduce the risk to product. These rapid and automated microbial monitoring methods may be adopted after validation has demonstrated their equivalency or superiority to the established methods. And I have mentioned rapid microdetection earlier. These rapid microdetection instruments for viable monitoring have been around for almost 15 years. And I was a distributor of some of these technologies while I lived in Australia. All I can say is that the concept is great, and applying these devices in aseptic processes has yielded limited results. However, there has been some more movement in that direction, and I would say keep watching this space. Again, the technology is very interesting, and you can get rapid detection. And the key is the validation of this technology and making sure that it demonstrates equivalency or superiority to established methods. Section 929, sampling methods and equipment used should be fully understood, and procedures should be in place for the correct operation and interpretation of results obtained. Supporting data for the recovery efficiency of the sampling methods chosen should be available. So understanding the technologies that you're using is critical. Luckily, here at Lighthouse Worldwide Solutions, we have an excellent knowledge center full of free information about all of our viable and non-viable products that deep dive into the technologies, so you have a good starting point when you use our products. As we also offer training courses, and our webinars are loaded with a lot of valuable information on applications and much more. And as I say, folks, we really go deep into the technologies. This is just an example that we talk about physical efficiency of our air samplers. We talk about the D50. You know, we understand our products, and we want you to understand our products, and we want you to be the smartest person in the room. And at the end of the day, you're the one standing in front of an auditor. So if you can impress that auditor with your knowledge of the technologies that you're using, all the better for you. You know, and we encourage that. As I said, we want to make you the smartest person in your room. So get onto our knowledge center and have a good look around there. There's plenty of good information available. Section 930, action limits for viable particle contamination are shown in the table. We notice here that in grade A, we're looking for no growth. So under A, settled plates should be exposed in grade A and grade B areas for the duration of operations, including equipment setup, and changed as required after a maximum of 4 hours. Exposure time should be based on validation, including recovery studies, and it should not have any negative effect on the suitability of the media used. For grade C and D areas, exposure time with a maximum of 4 hours and frequency should be based on quality risk management. Individual settled plates may be exposed for less than 4 hours. Under the notes for this table, contact plate limits apply to equipment, room, and gown surfaces within the grade A and grade B areas. Routine gown monitoring is not normally required for grade C and D areas, depending on their function. Just make a note of that. And under section C, you don't see it here in the screen, but if you do look at the actual Annex 1, you'll see that there's a couple of notes accompanying this table. I've just gone through A and B. Now, looking at C, it should be noted that for grade A, any growth should result in an investigation. Well, that goes without saying. If you do have a growth in grade A, you need to investigate where that growth has come from. And at the end, we have note 1 and 2, which I'll just talk about. And note 1, it should be noted that the types of monitoring methods listed in the table above are examples, and other methods can be used provided they meet the intent of providing information across the whole of the critical process where a product may be contaminated. Example, aseptic line setup, aseptic processing, filling, and lithization loading. Note 2, limits are applied using CFU throughout the document. If different or new technologies are used that present results in a manner different from CFU, the manufacturer should scientifically justify the limits applied and, where possible, correlate them to CFUs. Finally, under the environmental and personnel monitoring under viable particle, microorganisms detected in the grade A and B areas should be identified to species level and the potential impact of such microorganisms on product quality for each batch implicated. An overall state of control should be evaluated. Consideration should also be given to the identification of microorganisms detected in grade C and D areas, for example, where action limits or alert levels are exceeded, or following the isolation of organisms that may indicate a loss of control, deterioration in cleanliness, or that may be difficult to control, such as spore-forming microorganisms and molds, and at a sufficient frequency to maintain a current understanding of the typical flora of these areas. So this here really illustrates the contamination control in a nutshell. The contamination control strategy wants to merge multiple environmental monitoring data together. And I see this as a facility-wide approach. For example, let's say your cleaning agents are not rotated. Then I can see lower grade cleaners such as C and D increasing in contamination, and this contamination could be brought into grade A environments. And once you compile this data in a central contamination control strategy database, it will be easier to track such issues in real time and make faster responses to tackle the issues. Some companies take months to figure out their rise in fungi within their clean room, and it's down to cleaning agents not being rotated because this was not recorded or monitored. In fact, the issues came from the grade D environment and were transported into a grade A one where they showed up, and so the root cause investigation was focused in the grade A environment instead of where the contamination came from. And this is where we're headed, folks, and I believe digital technology and digital data transfers are the future of clean rooms in general. And this new annex really does have a lot in there, and it is a real call to action for all organizations to get on board and be ready for 2023. And if you need any help in these areas, Lighthouse here, we're more than happy to support and help you with the information that we have, the resources we have, the experienced people that we have that are available. So now we're going to move on to our final sections, which is Q&A. Okay, let's move on to our first question. Question number 1. Annex 1 now says that you must monitor throughout the production process, including setup time, whether your process is 30 minutes or it's continuous 24/7. You must monitor all of it. Yeah, the annex talks about making sure that you monitor during the process, like when you are aseptically processing the products and when the product is at risk of contamination. It talks about the setup time to monitor. It talks about the post process time to monitor, just so you can make sure that during or before you actually go into the processing, that you're guaranteeing that the environment is meeting grade A requirements. It also says that the cubic meter sampling is no longer enough, or spot monitoring throughout the aseptic process, particularly when we look at the viable monitoring. That's really important here, that there is a requirement now to monitor your viables continuously during the process, and it talks about settle plates and active air samplers. Now we do have portable units and continuous monitoring devices. Here we see the sample heads. They connect to vacuum systems, and you basically lift off the lid, put in the agar, the Petri dish. The same here with the portable. Take off the lid and add the Petri dish, and away you go sampling. You're going to capture the location, you're going to capture sample volume, the sample times, and in some cases with the right software, you can capture the operator as well doing the sampling. And I guess it's a bit of a shift in mentality, because a lot of companies were just getting a cubic meter sample for the viables to satisfy the standards. And but you must realize that there is a requirement in Annex 1 to capture all events. So continuous really is the way to go. So good question, and definitely look into the options out there for viable sampling with active air samplers and continuous systems. They are out there, and I'd recommend that you do consider them in relation to EN 1741 and also the obsoleted ISO 14698 standard, which is a good standard for selection of microbial devices. And there is a new ISO standard, I believe it's ISO 14644 Part 20, which again is in the draft format. It's not released yet. I'm waiting for that particular standard to come out and see if it is aligned with the old ISO 14698 standard on microbial sampling and selection of the devices, etc. Critical things to remember: the D50 to cut off the resolution of your air sampler, the ability of it to capture particles down to 1 micron is really important, the HEPA filtered exhaust, stuff like that. So definitely, viable sampling is becoming more and more required in aseptic processing. Question 2, Annex 1 now says you have to have zero growth, which calls for very high biological efficiency in monitoring equipment. Yes, we definitely want to, again, selection of the air sampling device, and I'm talking about the active air samplers here, which are on the previous slide. Definitely partner with a company that has validated the biological efficiency of their devices, and ensure that they have the capability of going down to at least 1 micron, and that's really important. And even the process of transferring your media onto your unit is also important as well, that you don't get any cross-contamination from the operators. But again, biological efficiency really talks about the ability of the air sampler to capture viable particles and render them viable. What I mean by that is that the impaction shouldn't kill the particles as they impact on the media, or the flow rate of the unit should be sufficient enough to capture right down to 1 micron. Question 3, Annex 1 does allow the use of settle plates for continuous monitoring. Yes, in some locations, environmental monitoring consists of an agar plate which is left open on a surface in the laboratory or clean room to capture any fallen microbes and gives a sense of the quality of the air. This is called passive monitoring, and in this case, the plate is known as a settle plate. The bugs that stick to the agar or on the plate during this process will form colonies and provide an indication of the air quality in the environment that you're monitoring. On the other hand, active monitoring, it's environmental monitoring, which is carried out using an air sampling or air monitoring device, and this is known as active monitoring. Air is drawn into the monitoring system and impacted onto the surface of an agar or a petri dish in a controlled way, and the subsequent number of colony forming units, CFUs, can be measured, and as the rate of air drawn to the system is also known, the number of CFUs per cubic meter can be established. So how do they compare? Not well for fans of passive monitoring. Active monitoring appears to be anything from 20 to 40 times more efficient than passive monitoring, because remember, passive monitoring, you're just capturing the particles that will settle onto these settle plates, hence the name settle plates. Whereas with the active monitoring, you're pulling particles that are more active in the environment, that aren't falling out of the environment quickly like you do have with larger particles. So passive monitoring using settle plates is an option in very low-grade environments where an indication of air quality is needed, but high accuracy or quantifiable measures are not needed. It's a low cost and easier to do than active monitoring, but it's less accurate measure of air quality. You're just getting those larger particles. So active monitoring requires specialist equipment, but provides a consistent, quantifiable, and more accurate measure of air quality. It is a preferred option for controlled environments and clean rooms up to grade A. And again, ensure that the unit that you're selecting is suitable for that environment. And again, I believe all of this should be considered in a risk assessment, and therefore you can justify which method you've chosen and why. And why might this be? Well, good question. All clean rooms have laminar flow air systems that create a downward flow of air. Typically, the airflow is around 0.45 meters a second, which means that on a 9 centimeter settle plate, around 175 liters of air is delivered every minute. We've been researching how this affects passive monitoring. We still are working on it. So far, one theory could be that the effective D50 value for a settle plate in a clean room with a downflow, as described, might be as high as 88 microns. So in this case, the settle plate is effectively 50% efficient at collecting microbes of size 88 microns and above. In the microbe world, 88 microns is a really large particle. And if you did have particles of that size floating around your clean room, you've got far more problems than the actual collection inefficiencies that we see here with settle plates. So I would definitely use settle plates within your clean room, but again, with a risk assessment, put them in the lower grade locations is what I would recommend, and save your higher grade locations and use your active air sampling devices, because you're going to collect a higher efficiency and more accurately what's happening in that environment. But again, piezo plates do have their uses, so don't get me wrong on that. Annex 1 does allow use of real-time systems, but be aware it says every event detected must be investigated. Yes. Now, this is a challenge for real-time systems, which typically detect many more events than an agar plate will grow, caused by items such as like even lab coat fibers, and even some pollens would register as a viable, but being non-viable and non-culturable in nature. These all need to be investigated. Remember, the table says 0 CFUs, or 0 growth. And these need to be investigated, and there are so many doubts about using this technology in the real-time environment. I say that with a little bit of experience, because 10 years ago, 12 years ago, I did represent such equipment, the real-time monitoring viable systems. And I believe the technology is getting better and starting to be introduced into the environment in aseptic processing. Whether or not it's going to really hinder or help, that is a question that we're going to be looking at over the next couple of years, as more and more companies are starting to go into this real-time data space using these real-time systems. So, and again, there is a vulnerability of biofluorescence and false positives. So I do think there is a place for this technology, but in a real-time system, monitoring in a aseptic manufacturing environment, especially when 0 CFUs are expected, and you need to do a root cause investigation for 1 CFU in the grade A and B environment, this could end up being more of a hindrance than a monitoring and control process tool, and particularly if you're having false positives on those biofluorescent triggers. There are units on the market that can detect using the biofluorescence technology and also capture on an agar plate the sample. So again, but you're limited to the 72 hours to get a positive on the CFU and identification of that species. So there is a place for real-time systems. I do see them in Q&A. I do see them in testing for operators who are gowning up to ensure that they're following the correct gown up procedures, and just to also detect if they are contributing to the bioburden in the clean room. So good question on that one. Question 5, in relation to 9.7, do you have any advice on how to locate the critical locations? Absolutely. In fact, we've conducted whole webinars dedicated to this topic. There is a lot to discuss, and I would direct you again to our knowledge center for the answers. Like, type in risk assessment and you'll see multiple papers and presentations on this topic. What I would say is that the risk assessment should be performed with a team of subject matter experts. If you look at this filling machine, there are mechanical, electronic, and SCADA systems, and this is a production machine used in a clean room. At a minimum, the team should have a process engineer, IT, production, utilities, and micromanagers present in all of their capacities to look at the critical locations. And also, if you do not have the in-house expertise, then hire subject matter experts who understand monitoring systems. Selecting an experienced monitoring systems vendor will also help you, as you can leverage their expertise. And we have helped many customers with risk assessments, URS development, and designs of monitoring systems, and the whole validations and performance qualification. So my advice is to start with the right team and develop a robust risk assessment, which will help develop a really good URS. And the URS will be the starting point and the finishing point of your complete project, and the traceability matrix will be built into that as well. So if you do have any questions, like, further questions, type them in, folks, and I will get back to you in a reasonable time. And my contact details are here. You can get me on LinkedIn. You can contact me directly with my email address. I've just put it up there on screen. So thank you all for participating today, and we will actually have our next webinar next month, and I believe Hashim will be coming again with a great topic. I'm not sure what the topic is at this stage, but stay tuned. We'll definitely get the information out to you, and stay on our website, our LinkedIn, our Lighthouse LinkedIn. You'll definitely get updates there. And just want to say thank you again, and I hope this webinar was informative to you. If you do have any follow-up questions, drop me a line on my email, contact me through LinkedIn, and I hope to help you out as much as I can. Take care, folks.